All right, everybody, we're talking proteins today. It's going to be great. Let's get into it. Amino acids are the subunit of proteins, just like nucleic acids are the subunit of nucleic acids of like DNA or RNA. So subunits of proteins are amino acids. In the last lecture, we talked about the pKa of different portions of amino acids, which is really the reason that we focus on that, why we even talk about buffers and pKa and pH and all that stuff. To kind of give us some background for understanding the basic structure of an amino acid. So there's an amino side with that nitrogen, right? There's a nice amine group there. And then on this side, we have our carboxylic acid side. Now, normally, it's relative, the pKa for carboxylic acid under physiological conditions, so the pKa of carboxylic acid is usually pretty low, like 2 or 3. And so it's usually going to donate its proton because it's more acidic than the normal pH of the cell or the blood or wherever you are. Whereas the amino group usually is 9 or 10, which is pretty basic for a pKa, and so it's usually going to have all 3 of these protons there. And so you're going to have, it has that extra proton, so it's slightly positively charged on this side. Well, it's got a positive charge. On this side, it's donated that, and it's got an extra electron, so we've got a negatively charged side. And we're going to be able to stick these together like little magnets that are going to stick together, okay? Basically, we're going to take a new amino acid and take its amino group and stick it onto this carboxyl group, and we're going to grow in this direction. Your R group can vary on its pKa, but yeah, it just kind of depends on what it is. If it's a methyl group, it's probably never going to donate a proton or accept one. But if it's a more acidic group or a more basic R group, then it's going to have a pKa that depends on the pH of the conditions that it's in, whether or not it has a proton. Now, these can act as buffer sinks for in the blood, and it's one of the minor buffer systems for the human body, but there are obviously bigger ones like the bicarb system, but it is substantial. And something to keep in mind, I'm going to bring this up a lot, so you're going to keep seeing this, is that the sequence of amino acids is controlled by the messenger RNA codons. And the messenger RNA codons is dictated by the DNA codons. So DNA makes RNA. Well, we make RNA from DNA. So we make messenger RNA from DNA, and we make our protein amino acid order from my messenger RNA, and the order of those amino acids determines the protein folding, and the protein folding determines the function. And so therefore DNA determines function, okay? But just with a lot of intermediate steps. Now let's talk about amino acids. I like this chart a lot. There's a lot going on here. It's a very informative chart, so you just kind of tackle one thing at a time. So you can say, all right, I want to know something about alanine. Well, it's aliphatic. It is not aromatic, so aromatics are like these ring structures, so we got some aromatics here. Is it acidic? Is it basic? Does it have sulfur? Is it essential? Is it non-essential? Which we're going to talk about just for a minute in just a second. But you can see these structures. I like these. I like this chart a lot because it the way they have the molecule structured is really nice. So you can see like 2 of them that I point out a lot are asparagine and aspartic acid. So they're kind of opposites. So asparagine has an amino group here, and aspartic acid has a carboxylic acid group here. So that's just, otherwise they're exactly the same. And the same with glutamic acid and glutamine. Amine, get it? And then the acid. Okay, so this has a carboxylic acid. Glutamine has an amine group here instead of a carboxylic acid. Okay. Anyway, but I just like this chart. It just gives you a lot of information. It has all the short little codes. Yeah, fun stuff. This is probably a better chart for actually memorizing the structures because it's a little more straightforward how they're actually drawn. They're less physiologically drawn. They're more drawn just straight, which is fine. So because you need to memorize all of the side chains, and you need to know all of the I will give you the names, but you need to know the prefixes, or not the prefixes, it's codes. So GLY and then the single letter code. So for glycine is G. Most of them are pretty straightforward. Some are a little weird, like tryptophan is a W. So I don't know what you're going to do. You just got to memorize it. There's 20 of them that you have to know. And the side chains are the most important. Now there's different ways. I mean, you can see here, there's different ways to organize amino acids. And this is probably the best way is based on the polarity of your side chains. So is it nonpolar? So just, it doesn't have any polarity at all. Is it slightly polar? So you can see you got a lot of oxygens here that are going to be able to form maybe some hydrogen bonding. And then you have your hardcore polar electrically charged. So these have full charges down here. So you got your acids down here. So they have a nice negative charge there, and you got your basics here, which have a nice positive charge. And so it just kind of depends on how you're looking at it. But this is, I like this chart a lot. This is what I based my quiz sheet on. So yeah.
Amino acids are made of carbon, oxygen, nitrogen, and hydrogen atoms, and some contain sulfur atoms. Selenocysteine is the only standard amino acid that contains a selenium atom. These atoms form an amino group, a carboxyl group, and a side chain, all attached to a central carbon atom. The side chain determines an amino acid's properties and is the only part that varies from amino acid to amino acid. Hydrophobic amino acids have carbon-rich side chains, which don't interact well with water. Hydrophilic, or polar, amino acids interact well with water. Charged amino acids interact with oppositely charged amino acids or with other molecules.
So I mentioned essential versus non-essential. So non-essential amino acids, the essential refers to consumption. So is it essential that you eat this amino acid? Well, that's a good question. Do you have the pathway within you, within your cells, or at least within an organ of your cells to actually make these amino acids? Because we need amino acids to do all kinds of stuff, either to make proteins or as precursors for other things. And so if they're non-essential, then you don't have to consume them from the environment. You can just make them yourself. They're just synthesized through a pathway. If they're essential, that means you need to be obtained from the environment, or they need to be obtained from the environment. So here we have the synthesis of non-essential amino acids, and they can go on and be used for all kinds of stuff. It depends on the kind of animal you are, but basically you can take proteins. If all you consumed in your diet was proteins, then you would be okay. You can convert proteins, you break them down and you can turn them into anything. You can turn them into sugar via gluconeogenesis, and you can turn them into basically all these other things. We got a lot of, we got serotonin, melatonin, all these messenger molecules. Anyway, so all kinds of stuff, but essential ones you have to consume in your diet. Now, there are conditionally essential as well. And conditionally essential are, we have the pathways to make them, but sometimes you need more than you're going to get from that pathway. And that's where you need to consume them. And so we have pathways for all of these, but if you're going through puberty or you're like a little kid, like one of my kids, they're pretty small, like 3 and 4, so they need a lot of protein in their diet. Albino rats also follow the same path. But essential amino acids, you need to consume those in your diet. So if you're human, you can obtain these from you don't have to be a carnivore, but there are certain animals that are obligate carnivores. So you may have heard about this, like cats can't really be on a vegan, quote, diet. They need to, basically it's the arginine. Taurine as well, that's a cysteine derivative, so you need to consume cysteine in order to construct taurine, which taurine's not really an amino acid. Anyway, so it's beside the point. So you need arginine, among other things. They have other, just like us, we have a bunch of essential amino acids that we have to consume, and so they do as well. But felids are not necessarily the only obligate carnivores. There's others. I threw minks, walruses, and crocodiles for no reason. There's others too, but like salmon and I don't know. There's other fish and stuff. Anyway, so lots of animals need to eat meat is the moral of that story. Now, moving on, there are a couple strange amino acids. Pyrrolysine, so it's like a lysine, but it's got a pyrrol group attached, or pyrrol. I don't say that right. You only see that in bacteria and archaea, not in eukaryotes. They have special enzymes called methyl transferases, so they're going to use that to move methyl groups, to transfer methyl groups. So what do you know? He's a genius. And so you can just, it's part of probably the active site of methyl transferases. But the more interesting one is in mammals, we have, and other organisms too, are selenocysteine. So it's arguably, quote unquote, the 21st amino acid. You will see some charts like these that will list selenocysteine. So here it even has a little reference down here. Selenocysteine is often referred to as the 21st amino acid, but it is encoded in a special manner. And so they're showing the codons here. That's what GGT, GGC. And so they don't list selenocysteine because we need to talk about how it actually comes about. But some charts do list it. I would argue that you don't need to list it because it's kind of a weird one. It's because you need selenium instead of sulfur sometimes. So that's what's kind of weird is we just happened to, long before we discovered selenocysteine, the body, before we named it, there's sulfur here. Cysteine is the sulfur, I spelled that wrong, sorry. Sulfur is the key component in the thiol group of cysteine. So there's your amino acid chunk there with your amino group and your carboxylic acid group, and then you have your, normally this would be sulfur if this was cysteine, and then in this case we have selenium replacing it. It's just a different element. It's not found in all organisms, but it is found in mammals, and it's really, really weird. So how we do it is normally you have a stop codon, so we're going to talk about codons in a second, but that should be something you're relatively familiar with. If you've taken microbiology, which you probably should have, but if you haven't, it's okay, we're going to cover it. The stop codon is UGA, but what you can do is normally you would just terminate at UGA when you encounter that stop codon. But if you take the messenger RNA and you perform post-transcriptional modifications on it, so you basically insert a weird loop region, like a stem loop region, into the messenger RNA that you want to make the protein that has cysteine. So you kind of twist your RNA really weird. Well, this is the RNA, but so you twist it up really weird, and that kind of calls in this enzyme, which is going to allow you to put selenocysteine in there. So it's really weird. There are lots of papers that talk about it. It's just kind of a strange thing. So understanding that RNA can have structure is important to understand how that works. So yeah. And one of the big things that we need to understand if you're going to take the MCAT, or you're just going to understand how things in life work, are the levels of protein structure. There are 4 big levels. I'm going to argue that there are sort of 5 because the supersecondary structures will kind of bridge the gap between the secondary structures, which is beta sheets and alpha helices, and into our tertiary structures. So they kind of act as domains and motifs that exist sort of binding these two together. So there's 4 big ones, though: primary, secondary, tertiary, and quaternary. So those are just terms you need to get in your head. Let's talk about primary structure. Primary protein structure is just the order of amino acids. Okay? So we start with our amino acid here. So we have a glycine here, and it has its amino end and its carboxylic acid end, and then we're going to tack on a proline and a threonine and a glycine and a threonine, and we number them based on our amino end, which is also called the N-terminus because you have that nitrogen there. So if you, in the literature, you'll see the N-terminal glycine or the C-terminal serine. And so this would be like the C-terminal serine, and this would be the N-terminal glycine. You'll see that nomenclature tossed around. So that's what that's referring to, because you have a nitrogen on one end, and yes, there's oxygen on the carboxy end, but I don't know, it's a C here and it's carboxy, so they call it the C end. I don't know, it's the C-terminus. It's just kind of got to accept it. And then you're going to number them based on the amino end. So you go 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, blah, 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 blah. That's the nomenclature for actually numbering the amino acids in a protein. But that's your primary amino acid structure. Now, when I take my first amino acid and I'm going to use a ribosome to build that new bond, that new peptide bond, because we call proteins also called peptides, or single amino acids can be called peptides, and then a protein itself is called a polypeptide, more than one poly, and then peptides being amino acids. So we can have a polypeptide, and so the bonds between our peptides or our amino acids are called peptide bonds. We connect those using a dehydration or condensation reaction where we're going to form water and then reform this bond. So this bond here, we're going to lose that hydrogen, and then this this ethanol group here is going to fall off. Or hydroxide group. Sorry, it's not. You understand what I'm saying. We're going to lose this. It becomes water, and then we reform the bond. Woo! Ribosomes do this, so that's what's happening when you actually have translation occurring. And so we've got our big subunit, we've got our minor subunits of our ribosome, the messenger RNA, 5' to 3' sticks in there. There's our transfer RNA, which is what bridges the gap between our messenger RNA and our actual amino acid. Okay, so there's our amino acid up there. And this is the actual portion between the P and the A site where we're going to reform that. So here's our NH. In this context, it's showing it's as NH2 there, and then we've got our C carboxylic acid with that OH. That H would be like right here. It's showing it. It's not there, but it's there. And so you're going to take this oxygen with a hydrogen and one of these hydrogens, and you're going to make water, which I'm not showing here, and then reform this bond. So this bond is going to get transferred to this nitrogen, and you're left with one of those H's falls off, and you got NH here, and you got C with the double bonded O, and then there's 2 more bonds to make it happy, and that is a peptide bond. Very, very fun. Now, the codons for the messenger RNA are what actually tell which tRNA to bind, okay, because it has an anticodon sequence here. And so let's talk about that a little bit. There's 2 types of charts that I like to, well, there's 2 types of charts that you'll see. You'll see some that look more like a table, and then you have some that look like this, these circular versions. I prefer this version. So if you're given a string of DNA, And every three nucleotides codes for an amino acid during translation. And so if you're given a piece of DNA, you say, okay, here's the codon sequence for my DNA. It's TTA. Okay, so I start here. I say, okay, I start in the middle, T, and I go to T, I go to A. Oh, it makes a leucine. Pretty cool. Now, let's try if I was given a messenger RNA and they give me the codons for that. What does GGG code for? Well, let's see. Start in the middle, G, 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 glycine. Cool. Awesome. And there's some redundancy built in. So if I had a mutation, actually, which we'll talk about later, and if you're in genetics, we talk about it a lot, GGA, GGC, GGU, there's a lot of redundancy built into that. Probably evolutionarily, you use a lot of glycines, and so you just want them to have some redundancy. So if you did happen to have a mutation, it wouldn't be a big deal.
Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Something I want to talk about is the connection between DNA and RNA and proteins. Since I'm talking about codons, it's really important because you're going to encounter this. I want to try and dispel some myths right now or some confusion that can happen. So this strand of DNA right here, okay, so there's my chromosome, I'm pulling this out, and there's my double-stranded DNA, awesome. I use my helicase, I open it up, but well, in this case you wouldn't use the helicase because we're doing transcription. Where you need to make messenger RNA, because messenger RNA is what we're going to send to our ribosome, and that is what is going to be converted into the protein, right? So we're going to make protein. So we need messenger RNA, and down here is my sense strand. So that is the piece of DNA that is going to have the codons that match up with the amino acid. So that's this codon chart right here, okay? And so what I want to do is I need a messenger RNA that looks just like this. Now it's RNA, so my T's look like U's, but other than that, it's exactly the same. So that's what we're going to see here. So UAG, TAG, UGC, TGC, CUA, CTA, right? I know it's confusing. It's CGG, CGG, right? So this messenger RNA looks just like the sense strand or the coding strand. I prefer calling this the coding strand. Sense and antisense is kind of an old school term. But hey, what you gonna do? So I like coding and non-coding because that makes more sense. So your messenger RNA, you want to look just like your coding strand because that's what's gonna code for it. And so this is referring to the coding strand. So if I give you these, that's what I'm talking about. I'm asking for the coding strand. Now, to actually make the messenger RNA, you have to make it off of the opposite, right? So if I want my messenger RNA to have a G, well, then I need to make it off of a C because that's what its opposite would be. And that's the antisense strand or the non-coding strand. And what's confusing to people is that that strand, yes, it's the opposite nucleic acid, but that's the strand that the RNA polymerase is going to use to sort of build its opposite side, right? So the messenger RNA is going to look like the coding strand, but it's built off of this other side. And so that can be a little confusing. You just got to kind of sit here and stare at it. But that's where you're going to encounter these terms. And so that's your antisense strand is what we code our RNA transcript off of. But it looks like the coding strand. Okay, so that can be a little confusing. Primary structure, that's just the order of amino acids, which is determined by the messenger RNA transcript, whose order of nucleic acids is determined by the DNA strand, right? So let me give you an example. I'm going to try and give lots of examples in this talk of specific proteins that fulfill each of the primary, secondary, tertiary structures, quaternary structure. And so I just want to talk about insulin. In this case, proinsulin is what I'm talking about, which is then matured into actual functioning insulin. It was the first protein whose primary structure we understood. And there's some disulfide bonds happening, which is pretty cool, between the cysteines to sort of hold it together. But how I want you to look at this is before we mature, so proinsulin actually has 3 chains, okay? And so we're going to start at one end of this chain. So I have a nitrogen, it goes, we keep going, we keep going this way, and there's my carboxylic acid. So there's my N terminus, and there's my C terminus. And what happens is we actually cleave off this C chain that I added on here that looks super high quality. Look at that, that's beautiful. And when you actually do the maturation process for insulin to go from pro-insulin to mature insulin, you remove that chain. And so you end up with 2 pieces. You have now 2 sort of chunks of protein that both have an N terminus and a C terminus. So that can be kind of confusing, but it used to be all connected by one little bridge there. And so this is what it actually folds into. And we're going to talk about the folding patterns. But you can see you get these little cysteine, these disulfide bonds here, which is pretty cool. So something else that I wanted to mention, because we just don't really talk about it a lot, is the evolutionary conservation, or lack thereof, that occurs in a lot of molecules. And insulin is one of those. So in other mammals like cows and pigs, the insulin molecule is almost identical, but that almost is really, really important, and it's really interesting. So you can see, if I go back here, we've got this cysteine connected to this cysteine, so we got a disulfide bond there, we got another one here, another one there, and we're seeing those same things. Here's that one kind of bridging that gap, and then you got 2 connecting these 2 strands, right? The A chain and B chain, or alpha, beta, whatever you want to say. I've seen it both ways, alpha, beta, or A, B. So in cows, they replaced this threonine and this isoleucine with valine. Okay, cool. Otherwise, they're exactly the same. And then in pigs, and also in cows at the end, we replace the threonine with an alanine. That's the only difference. So there's only 3 amino acid difference between cow insulin and human insulin. Pigs are even crazier. They only have an alanine here instead of a threonine. That's the only difference between pigs and humans. But that could be a huge difference. And so, yeah, it's just kind of fun. And so whether or not those are evolutionarily conserved is a great question. It's not a question. It's just, I mean, insulin works really well, and a common ancestor probably had the same one, and then for some reason cows have valine here, you know. But primary structure is what I'm really trying to get at. So the order from the N terminus to the C terminus. That's kind of cool. On cruising, so we're heading into secondary structure. So secondary structures are really, really interesting. There are really only 2 secondary structures, but we can do a lot with them and form some really cool things. And so there are 2 of those. There's beta sheets and alpha helices, or an alpha helix and a beta sheet. Sometimes it's called a beta pleated sheet or a beta strand. Beta sheet is probably the best way to talk about it, and then there's alpha helices. So let's talk a little bit more about them. So this is their structure. There's lots of pictures out there that you can find, but basically if we take our primary structure and we start folding those amino acids over on each other, there are a lot of amino acids that either have charges or that can form hydrogen bonds. And the order of those amino acids is going to determine the ability, or the sort of structure of being able to form hydrogen bonds, and how they're going to form, and at what angles they're going to form. And by changing those amino acids, I can thus change the kind of folding that occurs. And so if I have a specific set of amino acids, I can form an alpha helix. If I have a different set of amino acids, I can get them to more kind of fold over on these beta pleated sheets, and they could sort of form like a long flat piece. Yeah, it's kind of interesting. So let's talk about alpha helices first. So there's, usually we're only talking about a normal alpha helix. That's this one. Okay, it has 3.6 amino acids per turn. Okay, there are other helices, and so you might hear about them talked about sometimes. So there's 3.1s, which lo and behold has 3.1 residues, so it's a little bit longer. Okay, the 3 to 10 is another way that it's listed, but you can see it's a little bit longer. And then you also have pi helices, which have 4.4 amino acids per turn, so there's just more, so it's kind of a tighter turn. So you get your pi helix is kind of squished, and then your 3.1 helix or 310 helix is a little bit longer. Most of the time it's alpha though. In nature, very infrequently, you can find 3.1s, but in pi helices, you don't find those. Those are artificially generated in a lab. Though I say that, and then somebody's probably going to discover it. So, but hey, it's hypothetically possible. Now, helices occur in globular protein residues. We're going to talk about what that means when we talk about quaternary structure. But basically, you're going to see a lot of alpha helices throughout this, and we're going to see them depicted more often like this. or like this when we actually see some filling models. It's going to become very obvious in a little bit. And it's stabilized by hydrogen bonding between the amino groups on the inside and the carboxyl groups that are also on the inside. So they're kind of bridging that gap. So here's a little better way to look at it. So here's my N-terminus, here's my C-terminus. Doesn't really matter in this case. This one they're showing, of course, I said 3.6 residues. This one's showing 3.7. Whatever, it's fine. It's approximately 3.65 amino acid residues per turn. And you're going to have your R groups are going to stick. It kind of depends on the R group, but a lot of times they're going to face inside. And then to actually form the structure, you're going to have, there's my carboxylic acid. So look at just this gold looking dude here. So there's my N here, right? And then I've got my carboxylic acid here, that oxygen, and I can form a hydrogen bond between a different carboxylic acid group further up in the chain. Okay, so there's my carboxylic acid and another amino acid, and that can form a hydrogen bond here with this amino group. And then the opposite is also true. So this carboxylic acid can form a hydrogen bond with this amino group down here. And so that's how you form the alpha helix. Pretty cool. Now there are two kinds. So you can have left-handed and right-handed. It kind of depends if you hold your hand up. It depends on which way like the coil is coming towards you. So if it's right-handed, you hold your right hand up. If it's curling towards your fingers, then that's a right-handed helix. And if it's curling this way, it's left-handed. I don't know. Look at the chart. That's what it's here for. Left-handed helices are generally only in keratin and collagen, and so those are almost entirely just for structure. Whereas other alpha this is supposed to say all other alpha helices are right-handed, and so that's more of your for your globular proteins to form active sites. If you're not being involved with these are really long pieces, so long keratin and long collagen. And when we get into tertiary and quaternary structure, I'm going to talk about that more. But if you're not doing that, you're doing something else, and that's what these are for. So most of the time, you're going to encounter right-handed unless you're doing something structural. Now let's talk about beta sheets a little bit. And be parallel or anti-parallel. In the next slide, I'm actually going to show you what that looks like. So this would be anti-parallel because they're kind of flowing anti. If you went this way and then went really far and then came back again, it would be parallel. But it's not really super important. It's just there's different ways to kind of have your angles go. It's stabilized by hydrogen bonding. Once again, it's just sort of different. It's just structurally just a little bit different. So how they kind of fold over on each other, and that's how you get it. Instead of twisting around each other, they fold over. They're usually about 6 residues long-ish, and you don't usually have more than 6. I've seen a lot less, so 3 to 6 I would say is most beta sheets. You don't usually see more than that. You just don't. Sorry. So I got this from Draw It to Know It, which is an awesome website. I highly recommend you go there. They just look really nice. I don't know, and they're really high pixel counts, which makes them look really cool, and then I look great. They have really good study guides for this kind of stuff, especially if you're trying to do MCAT studying. They're really, really nice, and they go into some details that I'm not typically going into, like which amino acids comprised branched beta sheets, what are the aromatic amino acids, so they just have different ways to kind of learn those things. But beta sheets, you can have parallel or anti-parallel, so it just depends on the flow. So here's my N-terminus and my C-terminus, so if I'm going that way, and then I have another beta sheet that kind of folds over and goes this direction, see there's my N-terminus and my C-terminus, that would be anti-parallel. Whereas if I have them both going in the same direction, so you'd almost have to like have it go in this way, and then have it like kind of curve back around and then come back again the same direction. So I have my N-terminus, C-terminus, N-terminus, C-terminus, they're going in the same direction, that's parallel. And so you can see that's kind of what they're showing here. They don't have to look like this. Sometimes people get confused. They're just showing you that this would be parallel if you have N to C, N to C, and then this would be anti-parallel, N to C, N to C. So, but they don't always have to look like this. It could be all anti-parallel. It could all be parallel. It could be a mix. Who cares? Doesn't really matter. Anyway, I just like this because it has a good description of what parallel and anti-parallel is. About secondary structure and alpha helices and beta sheets, I wanted to show you an example just of an enzyme that happens to have both of those types in them. And so thiol groups are like on cysteines. That's when you have sulfur involved. So it's a sulfur with a hydrogen. I'm going to show you what that looks like in a second. but thioredoxin. So reduction means the gaining of electrons, right? And oxidation is the opposite. And so thioredoxin is an enzyme that is going to basically take two thiols and stick them together. Okay, so it can reduce, i.e. Add electrons to other proteins that have cysteine thiol disulfide exchanges. Usually with the CXXC motif, that's not super relevant. It's just it just happens to be like that's what you would have: a cysteine, then two random amino acids, and then another cysteine. And so they just like to do that. It's an antioxidant for, among other things. It can it's reducing thiol groups, and that can be good. It has 3 alpha helices. So we got, let's see if I can get this right. I think they're considering this 1, 2, and then there's like a little one here, or it's 1, 2, 3. I don't remember. And then a 4-stranded beta sheet. How could I possibly ask you a question about this? And then 4-stranded beta sheet. See, so we have 1 going this way, and then an anti-parallel. Oh, cool. And then we have another one here that's going anti-parallel to that one. And then we have another one. This is the bend, another one. But and then you have the parallel, right? So there's 4 of these. Where's this guy coming from? I don't know. Anyway, it's supposed to be 4 strands, but this picture is just being deceptive. It has a tertiary structure, which I'm going to mention later. It's called a thioredoxin fold, which sounds really cool. But I just want to talk about the activity of it because it's kind of fun. And so what it's going to do is, here's our substrate, here's thioredoxin, okay, and it actually has sulfur groups, and it wants to take its sulfur groups, and it's going to use them to, I'm sorry, we're breaking the bond between 2 sulfur groups. Derp. We're not making them, silly me. And so I've got my substrate with a disulfide bond, and I want to break that disulfide bond. So I need to do something with it, and so I'm going to take my sulfur groups that have hydrogens on my thioredoxin, and I'm just going to basically replace them. So I'm going to take this hydrogen, stick it on there. So this half of this bond kind of flips over here and sticks to this hydrogen, and then the other half sticks to this hydrogen, and then I form a new bond here, and then I have to restore it. And so the key to enzymes is that you can always restore them to their original state. And usually that's because with their own oxidation and reduction reaction. And in this case, it's dependent on NADPH, which we will talk about at a later date. That's going to come up later. It can reduce inflammation, reduces oxidative stress, blah, 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 blah. Who cares? It's kind of cool, though. Look at that. It's secondary structure. So because of the physical structure of the alpha helices and beta sheets, and the location of these 2 cysteines, so that's these 2 cysteines that it's talking about here, right? So because of their location and the structure of this, that allows it to grab onto the substrate, which is whatever protein we're trying to affect. Now we're moving on to supersecondary structures. So those are the structures that we're going to bridge that gap between secondary and tertiary structure. So tertiary structure is where we're going to sort of fold these beta sheets and alpha helices together. You're folding the folds. But that's sort of a simplification of how we actually typically do it. What we actually usually see are more supersecondary structures, like motifs and domains. And so I want to mention those a little bit. And so it's just more complex folding. So if I start folding combinations thereof, this is honestly one of the better pictures I could find. So if I take like a couple alpha helices and stick them all together, then that would be like a domain or a motif. So I'm going to try and define the difference between those. But so in this case, it's usually a domain is what we're typically referring to. And I will talk about that in a second. But so it's a supersecondary structure. So we're going to say, okay, if I have 4 alpha helices, which an alpha helix is a secondary structure, if I have 4 of those, that's an alpha helix bundle, and I can stick those together, and that forms either like a Rossmann fold is a really common motif that we see. So motifs are like repetitions that we see in nature, or the Greek key, which happens in antibodies or immunoglobulins. Lactate dehydrogenase, we're going to see when we talk about the citric acid cycle, and so anyway, so you can see if I start doing different combinations of secondary structures, then I get bigger things, and I can do stuff with them. There are a bunch of those. Thanks once again to Draw It to Learn It or whatever. There's lots of these cool ones, and so we see these a lot in nature, and so if we see them often, then we start giving them names. And so if we see like helix loop helix or coiled coils, we see that a lot in certain domains that will allow binding of DNA. Beta hairpin turns happen all the time. Greek keys are just really complicated. Beta hairpin turns. Beta barrels happen all the time. This structure should look super familiar because if you've ever seen like a cellular transport for like embedded in the cell membrane, they're usually beta barrels, or at least have some form of beta barrel structure. Sometimes you can, so that's like alpha helix motifs, beta helix motifs, or beta sheet motifs, and then if you start combining them, you can get weird stuff like zinc fingers. So zinc fingers are able to bind DNA, and you can find them in nature, and what we've done is take advantage of those, and you can make zinc finger nucleases, which will cleave DNA, and it's kind of like a CRISPR-y kind of thing, so you can modify DNA with them. Anyway, really, really interesting stuff. There's tons of repeats, tons of domains. So I wanted to talk about like the idea of what a tandem repeat is, because you're going to encounter that term. It's just a repetition of either a single set of amino acids. So if I have like 10 glycines in a row, that would be a tandem repeat because they're next to each other. So they're in tandem, but they're repeating. But you can have entire motifs that repeat as well. So like you can see all these, they're just repetition, right? So they're not polymers in that they don't continue on forever, but they do have repetition, right? So I can take this same subunit and then build it and build a larger structure, okay? And so that's just what a tandem repeat is. A domain is could either be made of a tandem repeat or it might not be, but domains are just if enzymes are capable of doing things, right? They lower the activation energy. We're going to talk more about that during the next set of lectures, but they do stuff, right? That's what enzymes' jobs are for. Well, the actual portion of the enzyme that does that is usually a domain, and there will be a couple different domains because enzymes are proteins and they have different jobs, right? So you might have like the business end of the protein, and that's the end that actually cuts something, like it's a nuclease, it cuts DNA. But then you need the other side of the protein might need to be able to bind to a scaffold or bind to another protein. And so it's going to have a different domain that lets it do that job. And so the domains are actually the pieces that are able to do the job. And domains are typically, like if we're thinking of tertiary structure, because we're kind of moving into that realm, that's usually what we're thinking about. Tertiary structure would be a couple domains stuck together to actually start to form things. So I want to give an example. So in this case, pyruvate kinase. Here's the structure of pyruvate kinase. We're going to encounter pyruvate kinase later. So pyruvate kinase is the last step in glycolysis. So when we take a glucose molecule and we chop it up, break those bonds, and we get some phosphates off of that. We have to put a couple phosphates in, but then we get 4 phosphates out, and we take those and stick them onto ADP to make ATP. So we make 4 ATP. And pyruvate kinase is one of those enzymes that actually makes ATP using substrate level phosphorylation, as opposed to oxidative phosphorylation, which is like the electron transport chain, and we're going to talk about all that stuff later. But this is one of those enzymes, and I just want to show you these domains. So there are 3 domains for pyruvate kinase. Well, there's technically 4 because you have the N domain, that's the N terminus, and then you kind of go through this whole thing, and then you end like right here. That's the C terminus. And so we name this domain because it just, I don't know, it just doesn't really do anything. And then you've got domain A is where you go first. It's in red. It's not actually red, but you know, whatever. And then you got your domain B, and this is where phosphoenolpyruvate, which is the precursor to pyruvate, is going to sit. And that's where it's going to bind. So this is the active site, is actually in between these 2 domains. We haven't really talked about active sites yet, but we're going to, and so we're going to encounter lots of enzyme active sites, and that's where that is. It exists between these 2 domains, actually, which is really, really interesting. Now, this enzyme, though, has 2 jobs, and one of the really cool things about pyruvate kinase is that it is one of the enzymes in these glycolysis cycle. Is it cycle? Not really. In glycolysis, that it is controlled by other products of glycolysis, okay? Which is kind of confusing. It's like meta level thinking. So if I make a bunch of fructose bisphosphate, okay, which is an earlier on step. I'm going to show where that is in a second. When I make fructose bisphosphate, if I make too much of that, it will bind to this portion, to the C domain of pyruvate kinase, and stop it from doing its job. So it's a way of kind of slowing up glycolysis so that I don't make too much pyruvate, because if I'm making too much, then I can get bogged down. I'd start building up toxic intermediates. It's just, you need kind of governors to sort of control how fast these processes are going. And so here's the diagram that I basically want you to memorize. There's fructose bisphosphate right here. So this is the byproduct from phosphofructokinase. It's going to take this guy and it's going to phosphorylate it. Okay, so it's a fructose with 2 phosphates, so it looks like this. And this molecule itself can bind right here. Okay, that's FBP is another name for it. It can bind right here and prevent this from working, which is really, really interesting. Now, that enzyme itself is actually all the way, number 10 right here, pyruvate kinase right there. It's this guy. And so what it's going to do is it's going to phosphorylate phosphoenolpyruvate. I'm sorry, it's going to dephosphorylate phosphoenolpyruvate. It's going to take a phosphate off of that and stick it onto ADP. So that's why you say, and it says 2 here because you have 2 of them actually going through the process, but it takes the phosphate off of phosphoenolpyruvate and sticks it onto ADP, making ATP. So that's how you get some ATP out of this, which is really, really cool. We're going to see this all again. We're going to talk about it more later, but hey, I'll let you be exposed to it now because then when you see it later, you're like, hey, I remember Dr. L talking about that. That's very cool. But these are the different domains. And so you can see domains are important. Pretty fun stuff. If that's our domains, we're moving slowly from secondary structures. We went through supersecondary structures. We went into domains, and that's where we're starting to really get into tertiary structures. And so tertiary structures, in a lot of contexts, if you're studying, we don't really deal with supersecondary structures. You just kind of lump domains into that because they are folding of folds, but a lot of times you can sort of, if you're folding those domains together, that's tertiary structure. It's still one continuous amino acid strand. Okay, we have not combined other strands together. So this is one giant long strand of amino acids. Yes, there's different folds, and we folded the folds, and we folded the folds of the folds, but it's still one continuous strand. And so talking about tertiary structure, it's just more advanced folding. So you can still have hydrogen bonds, of course. Usually hydrogen bonds are what's going to form your beta sheets and your alpha helices. But if I need to form bigger supersecondary structures, yeah, like domains, then that gets determined by either hydrophobic interactions. You can have van der Waals interactions if you have a bunch of hydrophobic R residues. You can form disulfide bonds or disulfide bridges, as they're sometimes called. You could have ionic bonds if you had like a glutamic acid and like an amine, like a glutamine or something binding here. But that's where you would get some more connections between these folds, and you're going to fold the folds. And this is really how you're going to generate the active sites to do things and to be able to perform these kinds of things. And so I wanted to give you guys some more examples. So in this case, we're going to move on to the citric acid cycle. Great picture of the citric acid cycle. And this one down here, isocitrate dehydrogenase, I moved this over. It's supposed to be on isocitrate dehydrogenase. So that is the enzyme that is going to convert isocitrate. Okay, so it's isocitrate dehydrogenase. So it's going to dehydrogenase the isocitrate. Okay, cool. It's going to make alpha keto glutarate because it's keto. We'll talk more about that when we talk about the citric acid cycle. But I wanted to show you the structure of isocitrate dehydrogenase. So it's still a tertiary structure, okay? And you can see it's very complicated. It's lots of folds. This is one continuous strand of protein, one continuous set of peptides. And if you zoom in on the active site, this is the active site within here that's going to actually bind to the isocitrate. So there's the isocitrate, it looks like that. And this is the active site within, I think it's right here, is where it's going to actually bind to that. But you can see there's a lot more going on because you have other jobs or other functions of different domains. So one of the domains is going to have the active site to actually dehydrogenate isocitrate, which is fine. You can see we have a lot of charged residues, which kind of makes sense. You can use the charged residues to sort of hold things in place. But then there's other portions of the main enzyme that we need to do other things. Maybe they need to bind to NADP, okay? Because we're going to take NADP and we're going to take a hydrogen off of this and form NADPH. Or it needs to bind to a control molecule. It just kind of depends on the functionality. So it's pretty cool stuff. What I like to talk about is myoglobin. So this is not hemoglobin. This is myoglobin. So it's a muscle-based protein that has heme. So this is your heme group. That's what actually physically holds onto oxygen. It is related, so it's structurally similar. Basically, if you take 4 of these, which I'm going to show you that in a minute, if you take 4 of these, you can actually form what is essentially a hemoglobin. So globular with heme. Okay, that's where the name comes from. But this is the heme. It's got an iron in there, which is cool. And that's the iron is positively charged, and oxygen has a negative charge. And so that's what actually grabs onto it. And so that's the structure here. So you can see this is a much smaller structure, but we still have tertiary structure because I've got my, it's like almost like one domain, but it has an active site. And that's usually what you're going to rely on with tertiary structure is I folded some of these folds, these alpha helices are folded on top of each other, and that would still be considered tertiary structure. Now you could argue, oh, it's like a supersecondary structure, or it's just a domain structure, it's not tertiary. Well, I don't know. They all kind of run together, basically, is what I'm trying to say. We need to talk about with the structures of our proteins, we're nearing the end, is quaternary structure. And that's when we're going to actually start combining amino acid chains. So we're going to combine full-on proteins. Now there's 2 big ways that we do that, that they actually form in the end. So what we've usually been talking about are globular structures. So they're really complex. You look at this thing or this thing, they're like little globs. Okay, that's where they came from. That's where that name comes from. So this is hemoglobin. So you can see hemoglobin actually looks like 4 of these stuck together. That's what hemoglobin looks like, and there's the heme groups. And so it's globular. That usually means it's water soluble because it'll keep its soluble portions on the outside. But there are insoluble forms, and that's usually our fibrous kind. So your fibrous ones like collagen and keratin, which can have like alpha helices that are left-handed that are rotating around each other. You can have multiple helices, and they just kind of keep on going, and they're usually structural. Not a ton of tertiary structure. You just got these 3 continuous chains that just sort of wrap around each other. Globular has a lot more tertiary structure. But it is separate amino acid chains that are stuck together. And a lot of times what you'll see is they'll be depicted as different colors, especially if you have repeating subunits. That's what you'll have here. So these are like 4 repeating myoglobin subunits that come together to form hemoglobin. How do you actually get things to stick together? Well, there's lots of different ways. You want, in general, you want a stable complex as opposed to a less stable one, obviously, though there are advantages to having a less stable complex. So I don't want you to, don't put emotions into this. So sometimes you want a stable complex, sometimes you want an unstable complex, because this would be easier to pull apart. So maybe sometimes you want to be able to pull it apart. Maybe you don't want to be able to pull it apart. It just kind of depends on what you're wanting to do. It's the exact same kind of bonding situation that you're going to have with the tertiary structure. So you can have ionic bonds, you can have hydrogen bonds, you can have hydrophobic bonds, you can have van der Waals interactions, you can have, there's another hydrogen bond, whoop-de-doo. You can have charge interactions, whatever. It doesn't really matter, but it's 2 separate amino acid chains stuck together. That's what's really important. Now, we call these oligomers, and they have different names based on how many repeating units there are. So if you had a polymer, which sometimes you'll hear that term thrown around, especially like in plastics or, I don't know, other industries like that, where you have a polymer of something. So a polymer is just a repeating subunit that just repeats forever. It just goes on forever. So it looks like this. When you have oligomers, there is some kind of limit. So you can have a monomer, which is just a single protein. You could have a dimer, which is 2 subunits stuck together. You have trimers, you can have tetramers, you can have pentamers, and hexamers, septamers, octamers, nonomers. They just keep on going. And so in the case of hemoglobin, it is a tetramer because it has 4 repeating units. Okay, so it would be a tetramer. Pretty cool. And so let's go back. We've got pyruvate kinase. I didn't lie to you, but I was kind of hiding this. So I just showed you 1 subunit, but actually we want some efficiency here. So we take 1 of these and we multiply it by 4. So here's 1 of them with its active site where it's doing its thing. And it's got the site where it can bind to FBP. And then we repeat that 4 different times. And so there's actually 4 chunks. It has a tetrameric quaternary structure, which, oh my goodness, that sounds so sophisticated. But we see that kind of thing happen all the time. And now it doesn't have to necessarily have repeating subunits. Sometimes we do. So in this case, this is flagella. So flagellin, it's like flagella for bacteria and stuff, or sperm or whatever. And so you'll have a repeating subunit. So in this case, you've got, this is your repeating unit, different domains. There's 4 different domains. Why did it go 0, 1, 2, 3 instead of 1, 2, 3, 4? I don't know. But hey, what you're going to do, toll-like receptor 5 is actually a detection mechanism from your immune system, it looks for toll-like receptor 5, which is only, or toll-like receptor 5 looks for this portion of flagella, which I actually did my grad work on. And so you can repeat that over and over and over again, and that's what actually forms the flagella, which when it binds at the cell surface is where the actual motor portion of that is. But this repeating subunit, you can see, keeps on going. And you can look at it like that. Look at that. That's very pretty. Whoop-de-doo. It doesn't have to be continuously repeating, so you can have really complex structures for quaternary structure. So in this case, this is ATP synthase. So we put a bunch of hydrogens on one side and we let them pass through, and that spins and then creates ATP. We're going to talk way more about that when we talk about the electron transport chain. This is the final enzyme associated with the electron transport chain, but it has a massive quaternary structure. Look at that thing. That is so cool. There's all kinds of moving pieces, like one piece doesn't move, but this piece does, and there's like a scaffolding for it. It's so cool. But you do have some repeating subunits. There's a couple alphas and a couple betas, but there's all kinds of other stuff. It gets complicated. Antibodies are another great example of quaternary structure. So we have 1, 2, 3, 4 repeating protein units. Okay, 2 of them look sort of similar for the heavy chain here. We got our light chains. Okay, so those are the constant portions of the heavy and the light chain are going to be similar. They're actually identical on this side. But then this region, so this whole region here and this whole region here are the same. And so they're coded from the same messenger RNA, so you have to think about that. That's interesting. And this is the business end. So this is the part of the antibody that actually binds to the foreign antigen. And so that needs to be able to be changed. Okay, so the physical protein structure here is what's going to physically bind to whatever it is. Now, oftentimes antigens are like let's say coronavirus is in the news, right? How could it not be? That would be like the spike protein for coronavirus may have a motif on its surface that binds into the protein antigen binding site right here. And how tightly that binds is really, really important. And it can have a lot of signaling that is connected to that. But you can see there's tons of, the quality isn't great, but you can see there's tons of beta sheets actually up here in the binding regions. And the actual structure, the protein, the amino acid structure here is an incredibly awesome and really cool area of research of how your body actually designs the protein structure here for which amino acids get stuck in there. You can have hypermutations as well, which is really, really cool. And, somatic hypermutation, meaning I can change the DNA, which you wouldn't normally want to do that in a cell, right? Why would you mutate yourself? But you can do that, and that's how you change what proteins are, or what amino acids are right here.
Primary structure. The primary structure of a protein is the linear sequence of amino acids as encoded by DNA. The amino acids in a protein are joined by peptide bonds, which link the amino group of one amino acid to the carboxyl group of another. A water molecule is released each time a peptide bond is formed. This linked series of carbon, nitrogen, and oxygen atoms is the protein backbone. Protein chains often fold into 2 types of secondary structures, alpha helices and beta sheets. An alpha helix is a right-handed coil stabilized by hydrogen bonds between the amine and carboxyl groups of nearby amino acids. Beta sheets are formed when hydrogen bonds stabilize 2 or more adjacent strands of amino acids. The tertiary structure of a protein is the 3-dimensional shape of the protein chain. This shape is determined by the characteristics of the amino acids making up the chain. Many proteins form globular shapes with hydrophobic side chains sheltered on the inside, away from surrounding water molecules. Membrane-bound proteins have hydrophobic amino acids clustered together on their exteriors, so that hydrophobic side chains can interact with the lipids in the membrane. Charged amino acids allow proteins to interact with molecules that have complementary charges.
So something I just want to briefly talk about because it's in my field. So I had to kind of relearn a bunch of biochemistry when I was in grad school because we were looking at HIV. So this is your T helper cell down here, and then this is GP120, or I'm sorry, your T helper cell would be up here. It would grab onto it. So GP120 is going to grab onto your T helper cell. And this is the virus down here. And embedded within the surface here of the virus is this region. It's called the membrane. Okay, because here's my viral membrane. Proximal, because it's close. It's close to the membrane. external, so it's on the outside because part of it's inside, part of it's outside, region. So the membrane proximal external region. And this is, if you zoom in, look at that, there's a nice alpha helix here. And these are the individual residues, okay? So you got a lysine here, and oh man, I can't remember all my residues. There's tryptophan there, and then you can see them sticking off. There's tryptophan, 666. And you can see these residues are sticking off. And there's a lot of consistency in here. And if you actually zoom in, you can see that if they stay constant, then we could use an antibody to actually grab onto it. The problem with HIV is that it mutates really well. And so what they found was that this region does not mutate super well, unlike most pieces of HIV. And so you can look in this region, and then you can take antibodies and see, you just throw them at this region and see if they bind. And it turns out there's 3 different kinds that bind. And let me show you a better picture of this. So here is that piece, okay? So if we're looking at this piece here, so if we zoom in on that, here's a chunk of that, here's a different chunk, and here's a different chunk. So these are all just chunks of this piece down here, okay? And so if I make that piece of amino acid, this is the antibody piece that would actually bind to that chunk. So in yellow is that piece right here. So that's where the antibody binds to. And there's in blue. So if you have a 4E10 is the name of the antibody that binds to that region. And the 10E8 is the antibody that binds to that region. And the 2F5 is the antibody that binds to this region. And those regions are consistent. So there's 2F5 and 4E10. Z13E1 is another binding region. But it's really, really consistent. So this is your amino acids. Okay? And so we can see 662 going to 683. So this would be our N terminus and our C terminus. Okay? Or there it is right there. N terminus, C terminus. And there's the region of the gene. So these are different genes, and that's the actual region. And these are the amino acids coded by that portion of the gene. So it's very, very interesting. I have another picture of it. It's just another way to look at the amino acids. So you can see you got here N, W, L, S, A. It's this, but it's just a different way to look at it. I just really liked this. This was the region that I was trying to make a vaccine with. So we took this piece out. So we took a piece of DNA that coded for this and put it into lactobacillus, which is bacteria, and then put that into mice and saw if the mice would make this antibody, the 2F5, which would bind to this part. So we were trying to make this antibody in mice artificially. And it worked. You would think we'd get more funding for it. Weird. So moving on to the last part of this talk. My voice is going, so I'll try and go fast. You were double timing this, or at least 1.5. I've talked pretty fast. So if you're double timing this, I'm talking real fast. We need to talk about how proteins actually get folded and how we get rid of them. Really, really important, okay? So chaperones are the name of the enzyme. Oh my goodness, that's scary. Okay, chaperones are what we're talking about. So chaperones are going to take an unfolded protein and help it to fold up together. Because sometimes proteins will just naturally fold together correctly. Great, awesome, they're going to do their job, sweet. So therefore, you don't always require a chaperone, but sometimes you do. It just basically kind of gives it a cozy little spot to just sort of fold properly. There are different chaperones. They're called heat shock proteins. So when we first discovered chaperones, what they did was they would take cells. What's the easiest experiment you could possibly do? You're like, okay, I have a cell in a petri dish. Let's heat it up and see what happens. And if you do that, and then you look at the proteins that come out, heat shock proteins start showing up. And it turns out when you heat cells up, the proteins that make up a lot of cells start to unfold because you're adding energy to them, right? So you can start to break those hydrogen bonds or the covalent bonds if it gets really hot, and they start to unfold. And so chaperones are heat shock proteins. So if I shock a cell, it starts making heat shock proteins. And HSP70 is the main one, and HSP40 is a secondary one. And they will just help the proteins kind of fold back into their proper shape. If they don't fold right, then you can get rid of them. But for the most part, that's what chaperones do. The actual process is far more complicated, just like everything in the body, but there's lots of different ways to kind of go. So here's my protein, I need to fold it. And proteins kind of go through different stages. So I could misfold it and it's broke, so maybe I need to break it down, or maybe I could form it into an aggregate. This is actually usually not a good thing. Amyloid fibrils are what cause like Alzheimer's or prion disease, which I'm going to talk about in a second. But if I have my regular protein, I can go back and forth between intermediates. Sometimes it gets unfolded. If things break, that's what this is talking about with chaperones. So you can see chaperones, there's different kinds. So these aren't the exact same one. But there's different types of chaperones, and they're going to just help your proteins fold, which is very, very fun. This was a higher quality earlier. Let me kind of shrink it down so it's nice. Chaperonins are another version that I just want to briefly talk about for like 10 seconds. You can time me. They provide better conditions. They're a little beta barrel looking protein thing. You put your protein in there and it allows it to fold better. So it's really not that different from this little guy here, but it's more like a little barrel, which is kind of fun. And you use ATP to make that. 1% of all cellular proteins, I beat, my 10 seconds are up, but 1% of all cellular proteins are chaperonins. That should not terrify you, but just put you in awe of, wow, a lot of proteins must need to be folded properly. Yes, yes, they do. All right, we're cruising on, we're almost done. How to actually destroy proteins. You may have heard of this process. I have a YouTube video here that you can watch if you want. It's not necessarily required, but I'll put it as an extra video that you can watch, or you can just click this. But proteins are actually destroyed. We ubiquitinate. If you want to read more about how to actually ubiquitinate and how we decide what proteins get ubiquitinated, you can go here. But ubiquitin is just like this little chunk of protein that you stick on there, and it marks it for death. And by death, I mean dissolution or cleavage, basically. So you're going to, if you have your protein and it's got a bunch of ubiquitin, and if it gets long enough, ubiquitin has other jobs too, but mainly its job is if you have too many ubiquitins attached, then this protein is then it then gets marked to be sent to the proteasome, which is a massive complex that will destroy the protein and break it into smaller peptides and eventually into individual amino acids so that you can make more proteins. So if you have a mistake starting to happen, well, then this guy's going to get rid of your mistake for you, which is pretty nice. So that's called the proteasome. Pretty cool.
The proteasome binds and removes the polyubiquitin chain and unfolds the protein. The protein is threaded through the proteasome chamber, where it's chopped up into building blocks to be reused for the synthesis of new proteins. The ubiquitin can also be recycled.
Pretty cool. Last thing I want to talk about is what happens when proteins go wrong. So prions cause prion diseases. So this would be like Creutzfeldt-Jakob. You can have spontaneous Creutzfeldt-Jakob, you can have infectious Creutzfeldt-Jakob. Mad cow disease is the one that everybody knows about. Kuru gets tossed around as well. Basically, there's this protein. It's got 3 alpha helices and a little anti-parallel beta sheet. So this is the little anti-parallel beta sheet there. It's a little cute little dude. It's harder to see on a smaller model of it, but you get that little guy. But basically 3 alpha helices. And then what can happen is we don't know what its job is. We've been studying it for years, and one of the guys on my committee was a really big researcher in here, and he was like, yeah, we still don't really know what prions are for, and he would know. But we know it's associated with lots of things, and it's this little protein. And what happens is in a neuron, you can happen throughout the body, but in the brain is where you see a lot of prion protein. And what can happen is you can go from form A here to form B. And so you can see you're converting some of your alpha helices into beta sheets. And what that does is it allows it to stick together and form these aggregates. So these aggregates then stack and into infinity. So they're forming a polymer, not an oligomer, but a polymer, and they aggregate together. And so they form what we call amyloid plaques, which isn't necessarily what this is. It's sort of a misappropriation of the term, but we still use it. And it can form plaques, or in the case of a lot of prions, they can form these super long fibrils. And you can imagine if it gets long enough, it'll just poke through the cell wall. They're really hard to destroy. Prions are notorious for, like, they'll resist a normal autoclave, like the kind that we use in the microbiology lab. That would not kill prions. You have to use a special autoclave. They're really, really dangerous, and they start killing neurons. And so you end up with what I call Swiss cheese brain. And we see this kind of plaque formation where you have these repetitions of these beta sheets. Those can occur in a lot of diseases, especially Alzheimer's. That's the most famous one. So if you're doing prion research, like mad cow disease, you actually get a lot of your funding from Alzheimer's research, which is really, really interesting. So that's proteins. Man, we've covered a lot. We went primary, secondary, tertiary, quaternary, peptide bonds, fibrils. Oh my goodness, we covered a lot of stuff. How to destroy things, how to fold things. That's a lot. It's a lot to take in. So I hope you paid attention and double timed this.